Good evening, one and all. Myself, Arvind Vithoba Kainde from MSc first year microbiology, SRTMU. Today I am going to conduct seminar on molecular biology. My topic is DNA replication. So let's discuss the basics of DNA replication. The DNA This is a complex process that takes place during cell division where by DNA makes copies, duplicates before the cell divides through mitosis and meiosis. DNA replication is a semi-conservative process where a parental strand, that is template, is used to synthesize a new complementary daughter strand using several protein elements, which include enzymes and RNA molecules. DNA replication process uses DNA polymerase as the main enzyme for catalyzing the joining of deoxyribonucleoside 5-triphosphates, that is DNTPs, forming a growing chain of DNA. Other proteins are also involved for initiation of the process and copying of DNA, along with proofreading capabilities to ensure the replication process takes place accurately. Therefore, DNA replication is a process that produces identical helix of DNA from a single strand of DNA molecule. DNA replication is an essential mechanism in enhancing cell growth, repair, and reproduction of an organism. DNA replication takes place in three major steps: opening of double stranded helical structure of DNA and separation of the strands, priming, priming of the template strands, and third one is assembly of the newly formed DNA segments. Now we will go towards model of replications. There were three models of replication possible for such a scheme. In conservative replication, two original DNA strands known as the parental strand would rebase rebase pair with each other after being used as template to synthesize new strands, and new the two newly synthesized strands known as daughter strands would also be pair with would also base pair with each other. One of two DNA molecules after replication will be all old and other would be all new. In semi-conservative replication, each of the two parental DNA strands would act as a template for a new DNA strand to be synthesized. But after replication, each parental DNA strand would base pair with the complementary newly synthesized strand just synthesized, and both double-stranded DNAs would include one parental or old strand and one daughter or new strand. In dispersive replication, after replication, both copies of the new DNAs would somehow have alternating segments of parental DNA and newly synthesized DNA on each of their two strands. So let's look out the structure of models of replication of DNA. So this is the model of DNA replication. This is semi-conservative, conservative and dispersive. To determine which model of replication was accurate, a seminal experiment was performed in 1958 by two researchers. Those are Matthew Meselson and Franklin Stahl. This is the mechanism of DNA replication. Uh, During the separation mechanism of DNA replication, during the separation of DNA, two strands uncoil at specific site known as the origin. With the involvement of several enzymes and proteins, they prime the strands of duplication. At the end of the process, DNA polymerase enzymes start to recognize the assembly of the new new DNA strands. These are the general steps of DNA replication for all cells but may vary specifically depending upon the organism and cell types. Enzymes play a major role in DNA replication. DNA replication because they catalyze several important stages of interprocesses. Uh, DNA replication is one of the most essential mechanisms of cell's function, and therefore intensive research has been done to understand its processes. The mechanism of DNA replication is well understood in Escherichia coli, which is also similar to that in eukaryotic cells. In E. coli, DNA replication is initiated at the auric locus, that is ORI capital C, to which DNA A protein binds while hydrolyzing of ATP takes place. The DNA replication enzymes and the proteins which are used are DNA ligase enzyme. This is the enzyme that joins DNA fragments together by forming phosphodiester bonds between nucleotides. Next one is hexanuclease. These are the group of enzymes that remove nucleotide bases from the end of the DNA chain. Topoisomerase. This is the enzyme that solves the problem of the topological stress caused during unwinding. They cut one or both strands of DNA, allowing the strand to move around each other to release tension before it rejoins the ends. And therefore, the enzyme catalyst, the reversible breakage, it's causes by enjoining the broken strands. Topoisomer is also known as DNA kinases in E. coli. Next one is telomerase. This is an enzyme found in eukaryotic cells that adds a specific sequence of DNA to the telomeres of chromosomes after they divide, stabilizing the chromosomes over time. Uh, then DNA, we have seen the first one is DNA polymerase. DNA polymerase are enzymes used for the synthesis of DNA by adding nucleotide by one to growing DNA chains. The enzymes incorporates complementary amino acids to the template strands. DNA polymer is found in both prokaryote and eukaryote cells. They both contain several different DNA polymerases responsible for different functions in DNA replication and DNA repair mechanisms. The DNA helicase enzyme, this is the enzyme that is involved in unwinding the double helical structure of DNA, allowing DNA replication to commence. This form two replication forks or each separated strand opening in opposite direction. So therefore, the helicase enzyme accurately unwinds the strand while maintaining the topography on the DNA molecule. The DNA primase enzyme, this is a type of RNA polymerase enzyme that is used to synthesize or generate RNA primers, which are short RNA molecules that act as a template for initiation of DNA replication. Further, now these are the enzymes and functions which are used in prokaryotic DNA replication. DNA pol 1, their specific function is exonuclease activity removes RNA primer and replace with newly synthesized DNA. DNA pol 2 repairs having repair function. DNA pol 3 have function as a main enzyme that adds nucleotide in 5' to 3' direction. Helicase enzyme opens the DNA helix by breaking hydrogen bonds between the nitrogenous bases. Ligase seals the gap between Okazaki fragments to create one continuous DNA strands. Primase synthesize RNA primers needed to start replication. Sliding clamp helps to hold the DNA polymerase in place where nucleotides are being added. Uh, so topoisomerase helps relieve the stress on DNA when unwinding by causing frakes and then resealing the DNA. Then the SSB, the single strand binding protein, binds to single stranded DNA to avoid DNA rewinding. So next are the steps of replication, DNA replication. That is the initiation, termination, and elongation. Initiation, elongation, and termination. First one is initiation. This is the stage where the DNA replication is initiated. DNA synthesis DNA synthesis is initiated within the template strand at a specific coding region sites known as origins. The origin sites are targeted by the initiator proteins, which recruit additional proteins that helps in the replication process to form a replication complex around the DNA region. There are several origin sites on which DNA replication is initiated, and they are all known as replication forks. The mechanism of DNA helix enzyme is by hydrolyzing the ATP that is used to form the bonds between nucleobases, thus breaking the bonds that holds the two strands. Additionally, during initiation, DNA primase enzyme synthesize small RNA primers that kickstart the function of DNA polymerase. DNA polymerase enzymes functions by growing the new daughter DNA strand. Next step is the elongation. The elongation, this is the phase where the DNA polymerase grows the new daughter DNA strand by attaching the original unzipped template strand and initiating short RNA primer. The DNA polymerase is able to synthesize a new strand that matches the template by extending the primer by addition for free nucleotide to 3' end. One of the template reads in the 3' to 5' direction, and therefore the DNA polymerase synthesizes new strand in the 5' to 3' direction, which is known as the leading strand. Along the template strand, DNA primase synthesize a short RNA primer at the beginning of the template in 5' to 3' direction, which initiate the DNA polymerase to continue synthesizing new nucleotides, extending the new DNA strands. The other template is 5' to 3' is elongated in an antiparallel direction by the addition of short RNA primers, which are filled with other joining fragments, forming the newly formed lagging strands. These short fragments are known as the Okazaki fragments. Later on, we are going to discuss about Okazaki fragments. Now, the synthesis of the lagging strand is discontinuous since the newly formed strand is disjointed. The RNA nucleotides from the short RNA primers must be removed and replaced by DNA nucleotides, which are then joined by the DNA ligase enzyme. The third one is termination. After the synthesis and extension of both the continuous and discontinuous strands, and enzymes known as discontinuous strands, an enzyme known as exonuclease removes all the primers from the original strands. The primers are replaced with the right nucleotide bases. While removing the primers, another type of exonuclease proofread the new strands, checking, removing, and replacing any error from during synthesis. DNA ligase enzyme joins the Okazaki fragments to form a single unified strands. The ends of the parent strand consist of repetition of DNA sequence known as telomeres, which acts as a protective caps at the end of the chromosome, preventing the fusion of nearby chromosomes. The telomeres are synthesized by a special type of DNA polymerase enzyme known as telomerase. It catalyzes the telomere sequence at the end of the DNA. On completion, the parent and complementary strand coil into double helical shape, producing two DNA molecules, each passing one strand from parent molecule and one new strand. So further, we are going to discuss about the DNA replication in prokaryotes. This diagram shows DNA replication in prokaryotes. Here the diagram shows as the DNA replication in prokaryotes. This is the replication fork is formed when helicase separates the DNA strand at the origin of replication. The DNA tends to become more highly coiled ahead of the replication fork. The topoisomerase break and reforms DNA phosphate backbone ahead of the replication fork, therefore relieving the pressure that results from this supercoiling. The single strand binding protein, that is SSB, bind to the single strand DNA to prevent the helix from reforming. The primase synthesize an RNA primer. The DNA polymerase 3 uses this primer to synthesize the daughter DNA strand. On the leading strand, the DNA is synthesized continuously, where on the lagging strand, the DNA is synthesized in short stretches called as Okazaki fragments. The DNA polymerase first replaces the RNA primer with DNA. The DNA ligase seals the gaps between the Okazaki fragments, joining the fragments into single DNA molecule. The replication fork moves at the rate of 1000 nucleotide per second. The DNA polymerase can only extend in the 5' to 3' direction, which poses a slight problem at the replication fork. As we know, the DNA double helix is an antiparallel, that is, one strand is in the 5' to 3' direction and other is oriented in the 3' to 5' direction. One strand, that is leading strand, complementary to the 3' to 5' parental DNA strand, is synthesized continuously towards the replication fork between the replication between towards the replication fork, because the polymerase can add nucleotides in the direction. The leading strand can be extended by the one primer alone, whereas the lagging strand needs two new primer for each of the short Okazaki fragments. The overall direction, as we saw in this DNA replication in prokaryotes, the niches that remain the niche that niche is formed that niche is remain between the newly synthesized DNA that replace the RNA primer and the previously synthesized DNA are sealed by the enzyme DNA ligase and catalyzes the formation of phosphodiester linkage between 3' OH end of the one nucleotide and the 5-phosphate end of the other fragment. So let's move towards Okazaki fragments as about this DNA replication in prokaryotes, the function of the enzymes, and all we have discussed. Now let's go towards Okazaki fragments. The Okazaki fragments, the two DNA strands run in opposite or anti-parallel direction, and therefore to continuously synthesize the two new strands at the replication fork require that one strand is synthesized in the 5' to 3' direction while the other is synthesized in the opposite direction, that is 3' to 5' direction. However, the DNA polymerase can only catalyze the polymerization of DNTPs only in the 5' to 3' direction. This means that the other opposite new strand is synthesized differently. But how? By the joining of discontinuous small pieces of DNA that are synthesized backwards from the direction of movement of the replication fork. These small pieces or fragment of new DNA strands are known as Okazaki fragments. The Okazaki fragments are then joined by the action of DNA ligase, which forms an intact new DNA strand known as the lagging strand. However, the Okazaki fragments are synthesized by the extension of RNA primers by DNA polymerase. However, the newly synthesized lagging strand is that it contains an RNA DNA joint, defining the critical role of RNA in DNA replication. The Okazaki fragment here. Now we go towards the replication fork. The replication fork and its function, as you can look at here, the parental duplex is there. Fork progression direction is given here in the diagram. Then the RNA primer is attached on the Okazaki fragment on the lagging strand. The replication fork is the site of active DNA synthesis where the DNA helix unwinds and single strand of the DNA replicates. Several sites of origin represent the replication forks. The replication fork is formed during the DNA strand winding by the helicase enzyme, which is exposed the origin of replication. A short RNA primer is synthesized by primase and elongation done by the DNA polymerase. The replication forks move in the direction of new strand synthesis. The new strand DNA strands are synthesized in two orientation, 3' to 5' direction, which is leading strand, and 5' to 3' orientation, which is lagging strand. The two sides of new DNA strand, that is leading or lagging strand, are replicated in two opposite direction from the replication fork. Therefore, the replication fork is bidirectional. So now we will go towards difference between DNA replication in eukaryotes and prokaryotes. The first point in eukaryote replication, due to the large size of eukaryotes, they possess 25 times more DNA. In prokaryotes, due to its small size, they have very minimal little DNA. The second point is eukaryotic cells have multiple points of origin and they use unidirectional replication within the nucleus of the cell. In prokaryotes, the cells have single point of origin and replication takes place in two opposite directions at the same time, and it takes place in the cell cytoplasm. In third point is that eukaryotes have 4 or more types of polymerase. In prokaryotes, prokaryote cells possess only 1 or 2 types of polymerase. Fourth point is replication of eukaryotic cell is slower, taking up to 14 400 hours. In prokaryotes, prokaryotic cells is faster, taking up to 40 minutes. In eukaryote, fifth point is that eukaryotes have distinct process for replicating the telomeres at the end of their chromosomes. In prokaryotes have circular chromosomal DNA, therefore they do not have any end any end to synthesize. Sixth point is that the eukaryotic cells only undergo DNA replication during S phase of the cell cycle, and in prokaryotes, the replication in prokaryotes takes place almost continuously. Thank you for listening me. So I will end my topic uh, on molecule. I will end my topic. Uh, Thank you very much.